Okay, I would like to invite all speaker. We come to the discussion at the moment. Can you please the three speaker to come to the stage? We will have a 15. Yeah, we have a 5 minute behind the schedule, but we start a bit late. So we will have 15-minute discussion. For the first rounds, I will invite two or three, three questions. Okay, Pak Pur, and then Bu Siti, and another one, Prof Sugeng. Okay, Pak Pur, please. Could you come to the microphone?
I have a question for Dr. Tejo. First is that viral load will affect the severity of the dengue fever? That is what question one. And question two, between NS1 and IgM and dengue plague, which one is the most appropriate? Which one is the most easier to be checked whether this is the case is dengue fever or shock? Which one of the three method? And 2 days ago, I find out 1-year-old baby with IgM positive. What is your opinion? Thank you.
Okay. Second question. Since the topic is very complicated, I think for Pak Tejo and Prof. Amin Subandiro, you may raise the question in Indonesia. Okay, Bu Siti, second question.
Assalamualaikum warahmatullahi wabarakatuh. Thank you very much. It's interesting presentation. I would like to ask to Pak Tejo. I wonder that there is no data from Yogyakarta, and my student in 2010 find that in Bantul, Yogyakarta, dengue virus 1 is the most serotype is dengue 1 from Bantul. It was done based on single tube RT-PCR using primer designed with Young et al. And in 2011 or 2012, my student no find serotype 1 in the mosquito in Bantul, but they do the based on two-step RT-PCR using Lanciotti primer. Why this is apa? Yeah. So do you can explain about this? Is this because of the primer design, or it is because of the different genotype? And I also want to inform that we have developed immunohistochemical assay using monoclonal antibody from in dengue of Universitas Gadjah Mada, that the sensitivity and specificity is about 94% and 90% using this study. And when we compare between secondary infection and primary infection, our result is sensitive both for primary and secondary infection based on immunohistochemical assay on blood smear preparation, thin and thick blood smear. Thank you very much.
Okay, third question, Prof. Sugeng.
Thank you very much for this opportunity. I would like to raise a question to Jeremy Brett. I heard that a number of researchers already developed experimental animal that's so-called humanized mice. Is that used also in Sanofi, Pasteur, and is it useful and valid for such an animal model for dengue infection and perhaps also pathogenesis? And to Profesor Amin, apakah juga saat animal model digunakan juga di LIPI ya? Mungkin Pak Tejo itu ya. Apakah humanized mice also used in LIPI and available also in IPP Bogor ya? Kemudian what is the ultimate parameter as the endpoint of dengue vaccination? Perhaps for the three speakers. Thank you very much.
Okay, Pak Tejo, you can.
Thank you. Thank you for the questions. I would like to answer a question from Prof. Pur. The first question is about the use of NS1 and IgG, IgM and plaque assay, if I'm not wrong. Which one is the most suitable for determining the dengue confirmation or the infection or severity? I'm not really sure, but each of the assay got their own what is called specific use. I mean, like the NS1 and IgM are quite useful in determining, and the NS1 especially determining confirm the early infection of the dengue. But the plaque assays usually only be used to for the research because it's quite laborious, expensive. Although it is quite the PRNT is the gold standard for the dengue titer determination, antibody titer determination. So each of them got the specific use, if I can suggest. And then the second question is about the severity and the viral load. It's already been established that in DHF, it is more severe dengue. The viremia usually higher than the one in dengue fever, so it's quite highly established. So lots of publication on that, and our data is also confirming that data. And then the third question is about the baby that got IgM positive. So it is possible, yes, of course, because IgM is the first 5 days of infection. Usually the IgM is produced by the body, so it's possible. And then the question from Dr. Siti, it's about the detection of dengue viruses, I think in Yogyakarta in 2010 and 2011. In 2010, dengue one is high in Yogyakarta, and the RT-PCR primer was used as Young et al. But the important thing here, I think, is for the detection of dengue virus. Because of the virus, it's an RNA virus, and then it's very highly mutated. Then the primers of the PCR that we use is actually a little bit different now. We already align our genomic sequence data with the Lancet primers, and we found 1 to 11 mismatch on the primers. So it is quite different now. The Lancet primer is already 2 decades ago, so 92 till now it's quite a long time ago. So with the evolution, with the virus evolve, then the primers become obsolete. It's not updated again. But then people keep trying the best to get the primers. So if you use the pan-dengue primers, it's quite specific, although it's not useful for serotyping. But yeah, it's the primer. Please be aware that the Indonesian dengue virus's genome is now quite different from the one described in the Lancet primers. So yeah, I have the data, so we modified the primers accordingly, and some of them got a better detection rate. And I think from Vietnam also developed quite good primers for the detection of dengue viruses in general, right? So Cameron Simon told me about that. It's quite now, yeah. Basically, now the technology of dengue detection is quite advanced with real-time PCR, and which is very sensitive, and then you know it's getting better now. And is there another question?
Yes, you got something on.
Okay, yeah. Another question is about the antibody sensitivities. What antibody said? Is it against NS1 or against NS1? Okay, yeah. It could be that the antibody raised by your lab is quite sensitive, but in my opinion, with the sample, with the lots of big sample number, then you will know exactly the sensitivity of and the specificity of your antibody to detect dengue infection, I think. But it's good because, as I mentioned before, the PenBio, it's PenBio ELISA, which is actually quite good. It's only 56% sensitive in Indonesia, and the BioRed ELISA, the Platelia, is only 40-something%. So in Indonesia, it's quite strange. The virus in Indonesia, the condition of dengue in Indonesia, is quite strange. So this, yeah, if you look at the PLOS entity, the author is there. So the BioRed is quite low. It's still lower than PenBio. Our data, which is in PMC infectious disease, will be published soon. Thank you.
Okay, Jeremy Brent, are you happy with the question? Is this?
Prof. Sugeng. So Prof. Sugeng, I think, asked 2 questions here. The first was about humanized mice, if that's correct, and then the second one was about an endpoint for vaccine development. So I don't know whether or not we have any specific experience with a humanized mouse model. But of course, in terms of dengue vaccine development, you have to know the strengths and limitations of whatever models you use. And so any animal model has to be rigorously validated to understand what aspects of human disease it mimics, and so what its use is. And it's possible, of course, that a humanized mouse model would help with the selection of vaccine candidates in the future. So it's something that needs to be fully explored. In terms of the endpoint for vaccine development, I think this can be very very simply stated as being a vaccine that safely reduces dengue burden.
Okay, Prof. Amin.
Thank you very much. I think I have two questions to respond. One is about the availability of humanized mice in PSSV Bogor. As you aware that PSSV Bogor only deal with primates because by nature is a primate center, but there are many other laboratories dealing with mice, but I'm not aware that they have already collection of humanized mice at the moment. The second question is, what is the best parameter for a successful vaccine, dengue vaccine? Based on the consideration of ADE, so first, of course, we need to have a vaccine that could elicit enough level of neutralizing antibody for those 4 types of dengue. The second one should be specific for Indonesia, of course, as already presented by Pak Tejo, that Indonesia has very specific characteristic of dengue virus. And the third one, of course, the stability of the vaccine itself, whether it's live attenuated or other type of vaccine. We should be able to make sure that the vaccine is stable enough. And number 4, of course, limited adverse event. Yeah, that are minimally 4 parameters that we have to fulfill so far. Thank you very much.
Okay, one more round for maximal 3. Okay, Prof. Scott, and then Prof. Ratman.
Well, this is not a question but a comment, and it starts with my being thrilled by Dr. Sasmono showing me that he was growing dengue viruses in primary human monocytes and macrophages. I have to give him the Halstead 2013 medal. Mostly when I go to meetings, people do what you did first, is grow dengue in renal cells. But I've autopsied more than 30 monkeys during the course of dengue infections and never recovered dengue virus from the kidney. So we tend to use these models, but how can we transpose the kinetics of anything from a model that actually is completely an in vitro artifact and not real? So congratulations on using, and I think you should do the kinetics of virus growth in primary human monocytes, because those are the cells that are growing dengue virus in real life. But what you forgot to do, so therefore I can't give you only a complete medal, a half medal, is adding antibody, because the kinetics of infection and the volume of replication and the innate immune response inside the cell is completely changed in the presence of antibody plus dengue viruses. But I think inadvertently you may have actually used antibody, because if you bleed somebody as a donor of primary human monocytes here in Indonesia, they're likely to be dengue immune. And what I discovered many years ago, and one of the problems of having done research many years ago is that my papers are way back there, 1970 or 1960, and I notice that nobody either reads them or cites them. But there's some very important information. And that is when we discovered the phenomenon of antibody-dependent enhancement, we discovered that if the donor was dengue immune, that you could not remove the antibody from the monocyte. So it's absolutely firmly attached. You can trypsinize it off, but you can't wash it off. So if your donor for the experiments you showed us were Indonesians, it's very likely they were dengue immune, and therefore, but maybe you know something about them that I don't know. At any rate, it's important to distinguish between a dengue or flavivirus immune donor and a completely susceptible donor. And if you want to do studies on antibody-dependent enhancement in vitro, you have to have susceptible donors, or else use human Fc receptor cell lines, THP1 or U937. I don't actually advocate that, because I think you're sacrificing the reality. For example, Mary Maravich has published papers in Journal of Virology showing that the innate response to immune complexes of monocytes and macrophages are different. So we have to be careful to retain that kind of specificity if we're going to learn about nature. But your experiment calls to mind another problem. That is that many people do research on dengue and attempt to make an in vitro model of something that happens in vivo, but they don't understand the kinetics of infection. Now, the clinicians in the room will tell you that dengue is really quite unusual for an infectious disease, and you have a silent incubation period of 4 to 5 days, you have febrile period of 4 to 5 days, but all of the dangerous things happen at the end of the fever. So when you defervescence. Now, I can't tell you how many papers I've reviewed in my life of people who model cytokine responses by sticking dengue in monocytes or whatever, mast cells, I don't care, and then after the infection, out comes all these cytokines. But the problem is that if that model is to be taken seriously, it means that right from the day the mosquito bites you, you ought to be producing those cytokines all the way through the infection. What would cause those cytokines to be produced in such an effusion at the end of the disease? So the experiment that I've seen that's published that I like was an experiment where somebody infected monocytes and then added human T cells directed against dengue antigens and added the two together, because that's sort of what we think may be happening at the end of the febrile period. But my final comment is about the use of terms in dengue. We're using terms carelessly, and the result is we're not thinking clearly. And if we don't think clearly, we'll never answer. It's like the chairman said, it's very confusing. Dengue is confusing enough, but if we use terms imprecisely. Now, the two terms that you use are virulence and virus load. Now virulence, I think if we're going to use the term virulence in pathogenesis research or in virology, we ought to recognize that virulence usually conveys an intrinsic property, the intrinsic phenotypic property to a pathogenic agent. And usually when we say virulence, we think that the organism causes something at the severe end of the spectrum. So I think we'd all agree that rabies is a virulent virus, produces severe disease and death in people of any age, irrespective of race or antibody status or anything. But dengue doesn't do that. If you say that dengue 3 or this strain or that strain or this genotype or that genotype is virulent, what you really are saying is that all by itself, that virus can cause dengue hemorrhagic fever, and not only that, but it will cause it again and again and again. My friend Dr. Leo from Singapore, and perhaps you here in Indonesia see adults that get dengue, and maybe they have diabetes or they have some other complicating problem, and they go on and get severe illness and maybe die. But that one case doesn't really make that virus virulent. So what turns dengue from being a mild disease to severe disease is antibody. And therefore antibody is a dependent variable. And we need a term that doesn't imply that the virus is intrinsically virulent. So I advocate using the term fitness. And when we do experiments where you have antibody plus virus, the amount of cytokine produced or the amount of virus produced or something might allow you to place that on a scale of saying less fit or more fit. But the word that I would suggest in your next lecture you use instead of virulence is pathogenicity. If a dengue virus can be used to infect completely susceptible people and lots of them, and most of them get sick, then I would call that a pathogenic virus. But I think virulence, which almost everybody uses virulence in the context of saying that's the virus that kills you. But killing in dengue is largely a dependent phenomenon, and I think we need to use a different term. But finally, virus load. Virus load is the gift of HIV to dengue. Now in HIV you have a chronic infection, and you can measure viremia usually by PCR, and it is a useful measure of how much virus is produced and how sick people are. But dengue, everything happens very fast. You have exactly 4 days or 5 days, but the day that you choose to measure viremia is going to be curtailed. The viremia is going to be curtailed by the amount of antibody that's produced. So the only studies in the literature that I think actually measure, quote, dengue virus load are the wonderful studies done by David Vaughan, who admitted children in the outpatient department when they had fever, 1 day of fever, and bled them for 5 consecutive days. And he actually measured the viremia in mosquitoes, which is a very fancy way of measuring virus. But what he described was if you had 2 days where the titer was the same, or 1 day where the titer was a little bit lower, and another day when it went up higher, and then finally the titer went down, that was peak viremia. So all the data we have in the literature that associates secondary dengue infection viremia or secondary infection NS1-emia are all based on these efforts to study patients on day 1 or day 2 of fever before the antibody response kick in. So I think it'll be less confusing forever if we don't use virus load sort of casually. Virus load really somebody should have gone to all the effort to make sure that you're measuring viremia in the absence of antibody. And then I think we'll all think clearer if we use critical pathogenesis terms more accurately. So anyhow, congratulations on getting started with human monocytes and macrophages. Please keep up the good work.
Thank you, Prof. Scott, for the comment. Okay, Prof. Pratman, brief question.
Thank you, Prof. Supargiono. Well, actually not really a question, but I would like to thanks for 3 speakers, because I think that Professor Supargiono already says complicated, yes, but we can synchronize from this research and also the consortium, because I would like to thank also with all Indonesian, institution. We have consortium is quite strong now, but I would like to remind after I learned from Pak Tejo, Dr. Tejo, and also from Dr. I forget your name, is Jeremy Brett, that we should share, I think, Pak Tejo, with the Pasteur Institute, I mean that from Jeff. So it's not so easy. We are combining between serotype and genotype. It's look complicated because in each season or each year there are different pattern of distribution, right? Genotyping. So this is not the task so for the consortium, if possible, the consortium itself, based on the new finding from genotyping, and which one we are going to develop for the vaccine candidate. I think we have to have review all this kind of finding and learn the experience from the Pasteur Institute. So it's not pessimistic but optimistic. You have a program for until 2020, right? 10 years. We should have the candidate of vaccine. But Maidan University already started 1993, right? 1993. How many years already? It's quite long time. But we have to speed up nowadays. And Pak Tejo, special for Pak Tejo, for the genotyping, I would like to suggest to continue the mapping due to the now seasonal because we have climate change and we have mobility of the people from one province, even one district to another district. That's all. Thank you very much indeed.
Thank you, Prof. Pratman. Pak Tejo, do you want to have a comment?
Okay, thank you. Thank you very much for Professor Harset of all of the suggestion. It's very, very inspiring for me actually to have your suggestion. For the donor of the monocytes, indeed it is immune. I mean, like got 4 antibodies against all 4 dengue viruses. And we already tried as well putting some antibodies, but since we use both of the donors in the experiment, then there is no big difference between the adding the antibodies to the viral load. So. Yes, I think so. We don't have any single sample of free of dengue antibodies, a single human. So we already screened around 20 samples in our institute, and no one got free dengue. They all got almost 4 antibodies by PRNTs. So, and also the virulence, the pathogenicity, that's quite inspiring as well. Yes, lots of publication use the virulence. That's why I also it's yes, it's good for us actually to learn about this from Prof. Halstead. Yes, I think, and then for Prof. Pratman, I think it's about the mapping according to the season, right? So, yeah, it's ideally maybe good to have data on it. So, but then sometimes this our surveillance here is only like it's because of our initiative, our own initiative to do this. So it's not the program. It's only based on our curiosity to have a dengue distribution in Indonesia, and then we are, it's not a program from the Ministry of Health. So because it's our own initiative, something like that. So why don't you give a program to us? Yeah, that's the thing, right? So it's very scattered research in dengue in Indonesia. So without the the unity in the research of dengue, the surveillance and so on, the data will be very, very, it's not solid because lots of cities got different dengue cases and so on. So, yeah.
Okay, I have a third notice from the organizer. We already have run out of times. We already heard about the presentation from Prof. Amin about the dengue research development in Indonesia. There are some research already done in Indonesia, many centers already involved, so that's promising. And from Jeremy Brett, already mentioned the preclinical research vaccine development that must be done to produce very effective and very safe vaccine. And from Pak Tejo already mentioned the variation, the huge variation of genetic and virus in Indonesia as well. So it means that we have to do more detailed research to because it may underlie the very complex immune response in dengue infection. So more detailed research need to be done in Indonesia, but we are in the right track, I think. Okay, give applause to the three speakers. Thank you very much, and the time's giving back to the organizer.
Okay, thank you so much, Prof. Spargionio and delegate speakers. Now I would like to request Professor Spargionio to please give the souvenirs to the speakers.
Thank you for all speakers and moderator. Ladies and gentlemen, this is the end of the presentation session. Before we continue to the next session, I would like to cordially invite you for the next 15 minutes, which have for the tea break which has been prepared outside in this meeting room. We will have an oral presentation at 11:15 after coffee break. And then for the oral presenter and moderators, we request you to please enter assigned room that have been announced yesterday. For the audience, we would like to request you to choose any of these floors for oral presentation. We have 3 rooms divided into 3 categories: clinical, molecular biology, and public health. Molecular biology on the 2nd floor, clinical in the 3rd floor, and for public health on the 4th floor. The oral presentation will continue for 2 hours till 13:15, and after that we kindly request all of you to have lunch directly, which will be prepared outside this auditorium also, and we will be joining in this room to continue the next presentation at 14:15. And thank you very much. May you all enjoy the tea break. Thank you.